Yet it is tempting, actually, not to speak after all of this, because there's no way I can compete with all the wonderful presentations, the thoughtful remarks, the incredibly generous, probably in some cases overly generous tributes. But I have to confess, I don't mind. I've enjoyed every one of them. You know, in a fortunate life, and mine has certainly been a fortunate life, there are nonetheless moments, peak moments, that you remember and will always remember, and this certainly is one of those. So thank to all of you for being here, for making that happen. I've loved every minute of this. I'm honored and thrilled that so many people are here, and so many people are here from long distances. I'll start out with some thanks, a little bit more extensive than last night, but with the same intent. First of all, thanks to Beth and to Mike for having decided that they'd like to lead such an effort, for talking to me to make sure I wanted it to happen, and yep, I decided I did. And to the organizing committee. Specifically, I've got my list here so I don't miss anybody. So besides Beth and Mike, Karen Keneally, Mingyao Li, Yun Li, Kathy Lynetta, and here at Michigan, Sebastian Zerlner, Dave Kabaki, Mandy Larson, Kyle Terwilliger, and Vera, for making this happen. And I know it's been complicated because honestly, the number of people that decided to come was sort of more than we initially anticipated. There was a need to move things around so that everybody could be together, and particularly everybody could be together at dinner last night. The thought that I was going to have to figure out 50 or 60 people to invite for dinner was daunting. And so the fact that a better place, a larger place could be found for that at last minute, thank you so much for making that happen, folks. Thanks also to Vera and the department and the U for supporting the event. The 11 talks have been just amazing, and so varied and so broad, and so exciting, so much good science, and so many kind memories that I truly will remember. And to my collaborators and staff and students and friends and family who are here, thank you so much for being here. Thanks especially to my wife, Betsy Foxman, to our sons, David, Kevin, and Richard, my sister, Barb, and lots of other family who are here. It is just incredibly great to have you here. I also want to mention my uncle, Galen Shorak, who was, for 50 years, a faculty member in statistics at the University of Washington. He actually celebrated his birthday yesterday, and he is now 87, and still, after my parents probably, the most important role model of my life, and one of my very best friends. When I started in Michigan in 1984, things were a little different, as Karen and others have described. In 1984, biology was the branch of science you went into if you didn't like mathematics, if you didn't want to have to do any math. Human genetics wasn't interesting. There was human genetics departments in a variety of places, including Michigan. And Jim Neal wasn't just the founder of the human genetics department at Michigan. He was really one of the 3 or 4 founders of the field, realistically, in the entire world. And statistical genetics was such a niche specialty, as Nancy and Steve and I can verify, that back in those days, you know, we could comfortably get together in one small room at the American Society of Human Genetics meetings each year, and we'd have 90% of the field in that one small room. One side benefit as a graduate student and a junior faculty member was we could know everybody in the field, and that's a slight exaggeration, but not a big exaggeration. And that was wonderful for someone getting started in the field. And to have those people who I've known so long to be represented here, that's particularly special. Mark, too, thank you. And so when I was looking for a faculty position as I was finishing my PhD, Ken Lang kindly sort of called around to his friends to see what might be possible, because there were exactly 0 positions open in statistical genetics that year. And I was really, really lucky that the faculty here at Michigan Biostatistics statistical genetics, that'd be okay. Maybe we'll try that. And never imagining, as a boy from the West Coast, that I'd live my life, or even a chunk of my life in the Midwest, and certainly not the rest of my life in the Midwest. But after having looked multiple times at different places, partly to make sure we were solving the two-body problem well, partly because opportunities came along that seemed like they might be interesting. When we decided not to move to Seattle in the late 90s, it was clear that we're Michiganians forever. It doesn't hurt that Kevin, our middle son, and his wife and 2 children live less than a mile from us, and Richard has now moved back here. So those are all reasons to stay, but even if they weren't here, we'd probably be here. So fast forward to 2026. Math and statistics and computing are central to the study of biology. Human genetics is, I think, one of the hottest fields in the biomedical sciences and has been for at least 10, 15 years. And statistical genetics is recognized as a high-need area that is central to what we do in genetics. For me personally, it's been an amazing 42 years. How is that possible? Well, I look at my gray hair, and I guess it is possible. But I've done research with outstanding people, wonderful colleagues who have become my friends. And I don't know how you get things better in science or in life than having work with people you just really enjoy. And yeah, life is too short to work with jerks. In fact, life is too short to work with anybody you don't really, really like. Yeah. And so together, we've developed statistical methods and computational tools used by investigators around the world. We've made strides in understanding the genetic basis of human health and disease. And best of all, as was just emphasized by the pedigree that shouldn't be sitting on the floor, but I didn't want to stand it up for fear that it'd fall down and break, the opportunity, the pleasure of helping to train an amazing group of scientists, 28 doctoral students, 13 postdoctoral fellows, and more than 160 students from our genome science training program. I look back on that and think, how blessed have I been to be able to do all of that. I'm so proud of all of you, and I will be watching, continuing to watch and enjoy and bask in your triumphs. And if you ever want some advice, don't hesitate to ask. And as I always make people promise me you'll listen, but you won't necessarily take the advice, because sometimes it's wrong. I've also had the opportunity through student admissions to interact with thousands of students around the US and around the world, and encourage well over 1,000, several thousands, to come to our graduate program at Michigan Biostatistics. I believe my most patriotic act as an American over my lifetime has been to encourage international students to come to the United States. Most stay and become incredibly productive members of society, and we've seen multiple examples of that over the course of this meeting. Certainly, we see multiple examples of that all the time. And the ones who go home help build bridges between societies, and I think that's really important as well. As it says in the tagline of my email and in the sign that I carried at the first Women's March after the election, the first time of the mistake in DC, immigrants do make America great. And thank you all who are for being here. Scientifically, it's also been an amazing 42 years. It's been really nicely described in several of the talks, so I'm not going to belabor it, but the dramatic improvements in technology and methods have made it possible for science fiction, actually several generations of science fiction, to become science reality over the course of this period of time. When costs go down and throughput goes up by sort of 6 or 7 or 8 orders of magnitude, science fiction becomes science reality really quickly. And we've seen that in genotyping and sequencing and lots of the other omics that we're doing. I tell my trainees, don't get your ego tied up too much in your work. But one place I do have ego tied up in my work is in the data sharing. The fact that we as a field really are collaborating, are sharing data, are sharing ideas, which is just as important as sharing the data. The fact that we as a field made that happen. I'm looking at Nancy, and I'm looking at Mark, and looking at lots of people here, Steve. That's been a real pleasure. Clearly, there's still a whole lot to do. And part of the reason I'm not really retiring is I don't want to be out of that. It's such a wonderful moment in all that's going on in genetics and human genetics and science that the idea of really retiring would mean not contributing to that in any way or not being involved. That doesn't seem sensible. Okay, so over the years, I've learned a few life lessons that I'm going to take a few moments to share. The first one is that life is a grant, not a contract. Do what you want to do it with the people you want to do it, and don't be afraid to change your mind. When we started the genome-wide association study for fusion, we had just gotten a grant funded to find map linkage peaks, an exercise that would have been futile. And Karen recounted very nicely and very gently the fact that we wasted a dozen years, and we didn't really waste them. We were doing the best that we possibly could with the available knowledge and the available technologies. But damn, I'm glad I had a grant, not a contract, so that we wouldn't have had to find map those false positive linkage peaks, and instead could talk with everybody we knew, including the people at the Center for Inherited Disease Research, who deserve a lot of credit, because we did, as Karen said, their pilot study for genome-wide association studies. And after having worked so hard in fusion to generate our first 1 million genotypes, then our first 2 million genotypes, and celebrated, it was champagne, I think, at least one of those times. In this small pilot study, we generated 800 million genotypes. Well, our colleagues did. And so we had the possibility to do work that otherwise, until then, was simply unbelievable. And then because Gonzalo and Yun and others developed genotype imputation, it allowed us not just to look at the 300,000 markers or so that we had, but to also combine with other groups, and then to look much more densely across the genome. And so technology has been incredibly important, but working together has mattered a lot. Okay, second, work really hard to be lucky. And that may seem counterintuitive because, of course, you're lucky or you're not. But the job for you is to weight the dice and put yourselves in positions where you're more likely to be lucky. If you need boxcars or snake eyes, that's a lot harder to get than a 7. So weight the dice so that you're much more likely to have a winning throw. And so go to places that are really good and interact, as Mark was saying, with the best kind of people in terms of being your collaborators. And treasure those relationships and make them go well as best you can. And go to places where if you do what you want to do and do it really well, people will love you. I feel very lucky that that's happened for me here. And do what you think is exciting and fun, because you only get to do this once. Well, I personally don't believe in reincarnation. Maybe it happens, but I have no empirical evidence on that point. So do what you want to do and enjoy it as much as you can. Third, in this highly interdisciplinary scientific world, nothing succeeds like successful collaboration. One of the great good scientific fortunes of my life is that Francis Collins and I arrived at Michigan in the same year, knowing nothing about each other, got to know each other through Bob Erickson in human genetics, and have been collaborating ever since. To have someone who's that great a scientist and that great a person, always available, always interacting, is unbelievable and truly fortunate. And since you're not always going to be successful, treasure your successes and celebrate your successes, large and small. And be resilient when you're not successful and realize that sometimes you just have to push forward. And then sometimes recognize it's time to cut your losses and do something else. And if genome-wide association studies hadn't come along when they did, we would have ended fusion, I'm pretty sure. And furthermore, if we hadn't had those annual in-person get-togethers where people ate and talked and sang and enjoyed being together, I don't think fusion would have lasted to the point of doing those genome-wide association studies. But the fact that we all liked each other, we worked well together, and we just flat out had fun being together mattered a lot. Okay. So, as I've said already several times, I don't intend to fully retire, at least not for now. Like I say, life is too good, this work is too exciting, the people I work with are too excellent and too much fun. But of course, I will be slowing down some. I will continue to do research, although probably no longer as a principal investigator. Sebastian has kindly agreed to take on the genome science training program, and it will be in very good hands. I hope to continue to be involved in student recruitment and student and faculty mentoring as long as people would like me to do that. I love those roles. And as always, I'm going to continue to do whatever seems interesting and impactful, and most of all, fun. I deeply believe in the importance of doing things that you just enjoy. Still, my move to emeritus status on June 1 does represent a transition. And I'm not sure it's been approved yet by the university. It's supposedly going to be decided sometime this week, so hopefully that will happen. But I can't imagine a more meaningful, special way to mark such a transition than what all you've done by being here for this event. I've got to stop. Thank you all very much.